Just a minute. Yes. So page number 15, we discussed the cell division, that is mitosis, yesterday. I'll just quickly repeat that. Right? Mitosis consists of or is divided in two processes, karyokinesis and cytokinesis. Now what is karyokinesis? Division of nucleus. Cytokinesis is division of cytoplasm. Right? Now in karyokinesis, the first phase or the first division takes place is of nucleus. So in karyokinesis, it is further divided into four phases: prophase, metaphase, anaphase, and telophase. Now you all know that what happens in prophase, in metaphase, what happens, everything is there in your book, right? Every point is there. In prophase, chromosomes become short and thin, right? Sorry, short and thick. Then two sister chromatids are remained attached at a small region known as centromere. Now this centrosome splits into two along with simultaneous duplication of centrioles. They form the aster, right? A star-shaped process or a star-shaped structure is formed, which is known as aster. This is in animal cells. An aster is not formed in plant cells. It is formed in animal cells. Now, the nuclear membrane disappears. Duplicated chromosomes start moving towards the equator. Now, in prophase, the chromosomes which are duplicated, as we all know that in cell division, during cell division, these chromosomes they get individuality, right? They are divided into two different chromatids, one chromatid and the other chromatid. So two different chromatids are there when cell divisions occur. So the duplicated ones, they start moving towards the equator, that means between the cell, that is in the center portion. Then metaphase. What happens in metaphase? Just have a look what happens in metaphase. In metaphase, each chromosome gets attached to spindle by its centrosome. Now this chromosome is attaching to the spindle fibers, right? Chromosome line up in one plane at the equator. In metaphase, they are lining at one plane in the equator. Like in the prophase, all the cells they just come into the center. Now in metaphase, they are lining up in the equator. They are forming a line. Then in anaphase, the centromere attaching the two chromatids divides. The two sister chromatids of each chromosome separate. Now these two sister chromatids, these again they get separated, and a furrow starts in the cell membrane at the middle in animal cell. What happens in an animal cell? So if we take animal and plant cell, in animal cell, the cytoplasm is completely divided by the formation of a furrow. Furrow means a bend, right? So in animal cell, the cytoplasm is completely divided into two cells, and in plant cells, it forms a plane, or it forms the line on the equator or the middle plane, and then it divides the two cells. Now this happens in anaphase, and in telophase, what happens basically in telophase, the chromosomes which are present in this or which are concerned with this, they again regain their shape, like from the shape where they started, that is the chromatin threads. They started from the chromatin threads, then they condense, and then during the cell division they form chromosome or they convert into chromosome. So in telophase, they are actually regaining their first shape, that is chromatin threads. Nuclear membrane is again formed. The cleavage furrow starts deepening in the animal cell. Like we all know, the cleavage furrow, it deepens in the animal cell and it completely divides the cell into two different cells. Now what is cytokinesis? Division of cytoplasm. As we all told right now, as I told you right now, this cleavage furrow deepens and splits the cytoplasm into two different daughter cells. Right? You can see the diagram. An animal cell is there. Right? Inside that nucleus is there, and then asters are there. The cleavage furrow deepens and it is splitted into two different cells. Right? Now come to cytokinesis, that is division of cytoplasm. That is the green topic, cytokinesis, division of cytoplasm. Yesterday we stopped at telophase. Now we will do cytokinesis. Now we all know at the end of telophase, just now we discussed what happens. Furrow deepens and the cytoplasm is divided into two. It is splitted into two. Thus, it produces two new cells. Now, karyokinesis and cytokinesis in that green box, all the nuclear changes that occurs during cell division are collectively termed as karyokinesis. As we all know, karyokinesis is the division of nucleus. So, all the nuclear changes which is happening or which is occurring during the cell division is known as karyokinesis, and it is thus followed by the division of cytoplasm, which is known as cytokinesis. So the first process which is taking place is division of nucleus, that is karyokinesis, and it is followed by cytokinesis. Now there are differences in mitosis in animal and plant cells. You can see the blue box, differences between mitosis in animal and plant cell. Now have a look at these differences. First, in animal cells, asters are formed. You can look at the diagram. You can clearly see in plant cells there are no asters formed. Asters means the star-like structure. So in animal cell, asters are formed. In plant cells, asters are not formed. You can make 2 to 4 differences with the help of diagram itself. You can see the diagram. There are asters present in the animal cell, and in plant cell, no asters are there. So first point is on the basis of presence of asters, right? Second one is cytokinesis by furrowing of cytoplasm in animal cell, and in plant cell by cell plate formation. We all know, we all just discussed that in animal cell, the cytoplasm is totally divided by the deepening of the furrow. It totally divides into two cells. So it is written cytokinesis happens by furrowing of the cytoplasm. That means deepening or this furrow deepens and splits the cytoplasm, right? But in plants, it occurs by cell plate formation. In plants, they are not totally splitting. They are forming a cell plate between the two cells, right? You can see the diagram on the above side. On the right hand side, plant cell, you can see one plate is formed. And by that plate, that is by plate formation, the cells are divided, right? So there is a difference in animal cells by furrowing of cytoplasm, and in plant cell by cell plate formation. Now third one occurs in most tissues throughout the body for growth and replacement. In animal cell. In plant cell occurs mainly at the growing tips for lengthening and for increase in girth. Now in animal cell, this mitosis in animal cell occurs for what? This is in the next point. For what purpose it is occurring? For the growth. It is written here, it occurs throughout the body. Main purpose is for growth and replacement. And in plant cell, it occurs at the tips. For what? For increasing the diameter. Girth means the diameter, right? For lengthening and for increasing the diameter. This is in case of plants, right? And in animals, throughout the body, it occurs for growth and replacement. Both have differences. Growth and replacement is different, and increasing and decreasing length and diameter is different thing. So this is the three difference between mitosis in plants and animals, right? So if in exam it may come, what is the difference between mitosis in animal and plant cell? You have to write these three differences. Now come to next page, page number 16. Now have a look at this green box, the terms parent and daughter cells. 1. The parent cell loses its identity in the process, and 2. Daughter cells are not necessarily feminine. So we usually use these two terms during cell division. That is, the parent cell splits into two daughter cells. But is are the cells produced really daughter cells? Like, are they feminine? No. We name them as daughter cells, but they are not really feminine in nature, right? We name them as daughter cell. It may be a male cell too. That means sperms are also present, right? Male gametes are also there, but it is named as daughter cells. So however, these two expressions are being used conventionally. Conventionally means these are socially accepted, right? Socially accepted by everyone. Now come to significance of mitosis. On page number 16, significance of mitosis. Do you all have any doubt till now? What is karyokinesis? What is cytokinesis? Any doubt?
No.
Okay. Come to significance of mitosis.
Ma'am, mitosis and meiosis.
Yes, what happened, Ayush?
Ma'am, mitosis and meiosis
Yes, so shall I
are,
explain it again?
yes, ma'am, I cannot understand that.
Okay, so I'll explain it again. Mitosis and meiosis are the two types of cell division, right? Clear? These are the two types of cell division occurring in organisms, right? In plant and animal cells, both. Now, what is mitosis? Mitosis is the first phase. Like, come to page number 13. Mitosis is there. Now, mitosis includes two steps, karyokinesis and cytokinesis. Now, karyon means a nucleus. So, karyokinesis will eventually be termed as or referred as division of nucleus, right? And cytokinesis means cytoplasm division. So, division of cytoplasm is known as cytokinesis, right? So basically, why we are dividing the nucleus and the cytoplasm? What is the need of dividing only these two? Why? Because these two are the living substances of the cell. We only divide the substances which are living. We cannot divide, or there is no point of dividing the non-living substances, right? These two things, that is the nucleus and the cytoplasm, they are the living substances. They are togetherly known as protoplasm. So these are the living substances. Therefore, the division is occurring in them. So in first phase, that is the first process is karyokinesis. First, the division of nucleus takes place. Nucleus is getting divided. Now this happens in four stages. There are four stages. These four stages are prophase, metaphase, anaphase and telophase. Is this much clear?
Yes, ma'am.
Yes. And now you have to look what happens in prophase, metaphase, anaphase and telophase respectively. Now before mitosis, you have learned that interphase is also there. So what is interphase? It is nothing. Here the cells just prepare themselves for the next mitotic division. They are synthesizing RNA and proteins and thus they are preparing themselves for the mitotic division. Now what actually the cell division is taking place in mitosis process, not in interphase but in M phase, which is the mitotic phase. Now what happens in prophase? You can have a look at the diagram. In prophase, centrioles are moving apart from each other. Chromosomes are distinct, means chromosomes are very clear. You just have to go through this diagram 2 to 3 times and you will understand the process, right? Now the sister chromatids, they are attaching to the centromere. You can see in the second diagram that in an animal cell, in the second round diagram, you can see the sister chromatids, they are attached by a centromere. Centromere is the middle portion which attaches two chromatids, right? This middle portion attaches two chromatids, like this is the shape of a chromosome. This middle portion where it is attached, where the chromatids are attached, is known as centromere. So they are forming a full proper chromosome, right? Now you all know, we have already discussed that during cell division these chromatids they just attach to the centromere and they form the chromosome. That means the chromatids condense, right? Just a minute. Yes. So where were we? In the next phase. That means the chromosomes, they are attaching to the chromatids are attaching to the centromere. That is the attaching point and they are forming the chromosome. These all happens in the prophase, right? Nuclear membrane gets disappeared. Why? Because these are dividing themselves. So the nuclear membrane is disappearing. Now come to metaphase. In metaphase, each chromosome gets attached to spindle. In metaphase, the chromosome is attaching to the spindle fibers which are already present inside the nucleus. You all know nucleus contains spindle fibers. So these chromosomes are attaching to the spindle fibers. From where they are attaching themselves? through the point of centromere. Now these chromosomes, they get lined up in an equator in metaphase, right? Then in anaphase, centromere attaching the two chromatids divides or splits. That is, in anaphase, what is happening? This centromere, which is attaching these two chromatids, it again splits, right? The chromosome which is formed, it again splits, and the whole furrow starts in the cell membrane and the middle in animal cell, right? That means the animal cell, it is totally divided through a furrow. That means a furrow is deepening inside it and it is totally divided, right? Then come to telophase. What happens in telophase? Basically in telophase, they are again regaining the normal shape. That means the chromatid threads was starting point. Then from there, these chromosomes were formed. These chromosomes during cell division, they were condensed, they were formed, right? So in telophase, they are again regaining their initial step. Sorry, initial phase or initial structure, they are again regaining. Yes. So in initial, they are again regaining the initial steps or the initial, what we say initial shape they are regaining in telophase. Now again the spindle fibers get disappeared and the nuclear membrane is formed. Why? Because the chromatids, when they were condensing, the nuclear membrane gets disappeared. But when they are again regaining their shape, the nuclear membrane again starts forming and the nucleoli also reappears, right? And the last phase is cytokinesis, where the division of cytoplasm is occurring. So cytokinesis is followed by what? Karyokinesis. After the division of this nucleus through all these phases, prophase, metaphase, anaphase and telophase, cytokinesis occurs. So it is the division of cytoplasm. Now in this is very simple. A cleavage furrow is formed at telophase. During telophase, when the last process is going on, a cleavage furrow is formed, a bend is formed. Now this furrow deepens and is finally cuts the or splits the cytoplasm into two daughter cells. So that's the whole process. This karyokinesis it's happening in four phases, and then at last division of cytoplasm is followed along with that, and in this the furrow is deepening very much, and finally it divides the whole cytoplasm into two daughter cells, right? But in plant cell, it is little different. In plant cell, a plate is formed. In animal cell, the cytoplasm is totally divided. In plant cell, a plate is formed between the cell and they get divided. We have we have noted the difference also. Like come to the page number 15, animals and plants, difference between mitosis in animals and plants. You can see the second point, cytokinesis by furrowing of cytoplasm. And in plants, cytokinesis by cell plate formation. So there is a difference between two. In animals, it is a deepening or it is by the furrowing of the cytoplasm, deepening of the cytoplasm. But in plants, they are forming a cell plate and they are getting divided, right? Have you understood, Ayush?
Yes, ma'am.
Yes, just go through these points. Do not go through the blue points, right? Just go through these points in page number 14. That is from interphase, prophase, metaphase. These are short points. So you have to remember what happens in prophase, then in metaphase what is happening, anaphase and telophase. Telophase is basically nothing, but they are regaining their shape. And from prophase to anaphase, they are forming the plate and they are arranging themselves, they are attaching themselves to the spindle fibers. All these things are happening from prophase to anaphase. And in telophase, they are again regaining their shape, right? So if you read this paragraphs 2 to 3 times, it will be very easy for you to understand. Also look at the diagrams because there is a difference between animal and plant cell, right? Just a minute. Just a minute. Yes, so you all have any doubt in this? This is little complicated. You have to remember what is prophase, metaphase. This is all about learning. So you have to just read it 2 to 3 times and it will be clear for you. Just go through the diagrams. Diagrams, it is more clear, right? So do you have any doubt? Do you all have any doubt?
No, ma'am.
Okay, Ayush, is it clear for you?
Yes, ma'am.
Right, just go through the sentences and just go through the diagrams. Also go through the differences, that is difference between mitosis in animal and plant cell, so it will be more clear to you, right? So in animal cells the process is little longer, right, little complicated, like the furrow formation and the deepening, it's little complicated in animal cells. But in plant cells it is not that complicated because asters are not formed and these only move towards different planes and they arrange themselves in a plane. So that's why it's not more complicated in plant cell, right? So an animal cell, it's little complicated. Just go through this once. It will be clear for you. Now one more thing. Is interphase, which phase is the longest phase? Can you all answer? Can anyone answer? Interphase or M phase? Is it interphase or M phase? Which is the longest phase? This is a very tricky, very confusing question. Can you answer? If not, then I'll answer. So, you may be thinking that interphase is the resting phase. We all have studied that the interphase, it's a resting phase, right? It is in resting, all the cells are just preparing themselves for the next cell division, right? This is the resting phase, they are synthesizing RNA and proteins. You must be thinking that way, but it is not true. Basically, if we say between interface and M phase, interface is the longest one. Now how it is the longest phase? While if you see in the diagram also, if we see mitosis process, karyokinesis and cytokinesis, everything is occurring. So you may be thinking that M phase is the longest phase, but in actual interphase is the longest phase. Now how? It is because we all know what is happening here. A cycle is continued, right? First interphase is there. All the G1 synthesis and G2 phase is happening there. And then the cells are entering into mitotic phase, the division phase. And then again the cells, newly formed cells, they are again entering into interphase. So basically interphase is coming between two mitotic phase, right? Interphase is occurring, then mitosis, then interphase, then mitosis. So between two mitotic phase, interphase is coming. So obviously interphase will be the longest, right? Interphase is coming between the two mitotic phases. That's why it is the longest phase in this cell division, right? So you all get the point?
Yes, ma'am.
Yes, ma'am.
Yes. So come to page number 16. Page number 16. Significance of mitosis. Significance of mitosis. Now, what are the significance of mitosis? This is very clear. You just have to look at the bold portions. Growth, repair, replacement, asexual reproduction, and same chromosome number. Right. Now, how it is significant or how it helps in growth? What is mitosis? A type of cell division. Cells are dividing, right? So by dividing cells, obviously if cells are divided, they get more in number. Obviously they will help in growth of the organism, right? Larger the organism, larger is the cell. So when the cells divide, when they get more in number, obviously they will help in the growth of the organism. Like for the cells to occupy the spaces, the organism's body will also grow along with that. So since it helps in the growth, so since this new cells are formed in the tissues, they occupy more spaces and therefore they help in the growth of an organism. Is that clear for you?
Yes, ma'am.
Yes. Now come to repair. Now how it repairs? It repairs repair of damaged and wounded tissues by renewal of the lost cells. Now repair is also done by mitosis. How? Because it is because you all know it just destroys, or just when the old cells become very old and become functionless, they are replaced with new cells. Now these new cells, they help in the replacement of the lost cells, right? So they basically help in repair function. Then replacement, we all know old and dead cells are replaced with the new ones. During cell division, new ones are already forming, right? When mitosis is occurring, new cells are formed. So these new cells replace the old and dead ones. Like for example, epidermal cells of the skin. Epidermal cells of the skin, these do not these do not function effectively for a long time. These have to be replaced from time to time, right? So this is the example, that is the epidermal cells of the skin. Now fourth is asexual reproduction. How? Asexual reproduction happens where? In unicellular organisms like amoeba, right? So they divide into two. Asexual reproduction means dividing into two. So actually the cells are dividing into two, so they also help in asexual reproduction. Then also maintains the same chromosome number in daughter cells. You all know in mitosis, first what happens? The cells are just growing, synthesizing RNA, and then they duplicate the DNA, right? I told you why they duplicate, because both the daughter cells must equally get the DNA molecule. So the same chromosome number is maintained in the daughter cells which are formed by the parent cell, right? They get equal amount of DNA, equal amount of same number of chromosomes up there. So this is the significance of mitosis. All these five you have to learn. Now come to, yes, now we have meiosis. So we have 10 minutes more. We will do the meiosis portion also. Now what is meiosis? Mitosis, you all know, cell divides and forms two types of daughter cells, right? Diploid cells are formed. We have already discussed in mitosis, diploid cells are formed, and in meiosis, haploid cells are formed. What do you mean by haploid cell? That means 4 in number. First, the parent cell divides into 2. Then these two newly formed cells again divides themselves into 2. At last, 4 cells are formed. So this is meiosis, also known as reduction division, right? Reduction division producing gametes. Now what are gametes? The sex cells, sperm and egg. These are the gametes. So meiosis is basically the reduction division. Now you will think, if meiosis is producing 4 cells, 4 haploid cells, why it is known as reduction division? Now you will get to know why this is known as the reduction division, because since they are forming 4 haploid cell, but what happens in mitosis? In mitosis, the same number of chromosome is maintained. But in meiosis, when they are forming 4 cells, 4 daughter cells, the chromosomes get halved. Like each of the daughter cells get half number of chromosomes. So because of this lessening of chromosome number, or the halving of chromosome number, this is known as reduction division. Why? Because at last, the 4 daughter cells, chromosome number is just the half of the parent cell. But this is not same with mitosis. In mitosis, chromosome number is maintained. The chromosome number is parents is the chromosome number in daughter cells. So it is maintained there. But in meiosis, it is just halved, right? Now meiosis, come to meiosis. We already know it's a reduction division. It produces new gametes, that is sperm and egg, also known as sex cells, right? And in flowering plants, where does this, where are they produced? Like the gametes, the sex cells, where are they produced? In the anthers. Anthers are the reproduction organs in plants. So in plants, they are produced in anthers. Now, number of chromosome in the sex cells is halved. As I told you, in meiosis, when they are finally formed into 4 haploid cells, that is 4 in number, haploid cell, the chromosome number is just halved. It is reduced, right? That means the daughter cell will only contain half the chromosome number of the parent cell, right? Now, for example, we can take out of 23. You know, how many pairs of chromosomes are there? 23 pairs, right? So this is for your knowledge, I'm telling you. 23 pairs of chromosomes are present in a human body, right? Now from 23, only one chromosome is the sex chromosome. That means the one chromosome is for the gametes. The other 22 are known as with other name. What is that name? Autosomes, right? The other 22, the first 22 ones are the autosomes. These autosomes, what they do, what is the function of autosomes? Autosomes do the function of inheritance. Inheritance means you all know the characteristics of the parents is passed or transferred to the offspring. So that is inheritance. So these autosomes, that means these 22 pairs, these helps in what? These helps in the transfer of inheritance from parents to offspring. And there is one left, which is the sex chromosome, and these sex chromosome identify the sex of the organism, right? Is this much clear?
Yes, ma'am.
Okay, now come to significance of meiosis. Also, in meiosis, this is essential because when the male and female gametes fuse during fertilization, the normal double diploid number of chromosome is reacquired. Now, when we write or when we calculate the chromosomes number, we have a rule like diploid number is expressed as 2n. 2 means diploid, and the haploid number as n, right? It is not written as 4n. Haploid rule is n, and the diploid rule is 2n. So you can see this extra diagram given here. This is written here extra, not in the syllabus, but you can see 2n, which is a diploid number, right? Again forming a 2n. Again it is forming a diploid. Then this 2n is splitting into 2. N refers to haploid. That means this diploid is forming haploid cells. And then again it is forming 2 haploid cells. So at last 4 haploid cells are present, right? So this is for your, just for your understanding, right? You can see the first division, reduction division, that is mitosis 1. Chromosome pairs are separated. Each daughter cell receives only one chromosome from each pair. Now mitosis 2, second division, centromere is splitting. The chromatids of each chromosome separate to become independent chromosome. So for haploid number, they are just separating their chromatids. They are becoming individual chromatids, right? The chromosomes get separated, right? So this is for your better understanding. So meiosis, you all know, is a reduction division. Why it is a reduction division? Because the cells formed, the daughter cells formed at the last, they only contain half the number of chromosome of their parent cell, right? That's why it is known as the reduction division. Now come to significance of meiosis. Yes, we have 5 minutes. Significance of meiosis. So we have already known about the significance of mitosis, growth, repair, replacement, asexual reproduction, and same chromosome number. But in meiosis, the first point is chromosome number is halved. As I told you, the chromosome number is halved in the daughter cells. It is not same as the parent cell, right? So that on fertilization, the normal number is restored. How the normal number will be restored? The chromosome number is already halved in the in the haploid cells. When these cells fuse during fertilization, obviously, when these cells with half number of chromosome, they are fertilizing, they are fusing, then again they are restoring the actual number, which is 2n, right? 2n number is restored during fertilization when these gametes fuse together, right? And when they are divided, the chromosome number is halved. It provides for mixing up of genes. Mixing up of genes is also done with meiosis. It happens in two ways. The first is the maternal and paternal chromosome get mixed up during the first division as they separate from the homologous pairs. What happens in the first division? The maternal and paternal pairs get mixed up. That means one maternal pair, one paternal pair, they get mixed up and form a chromosome. What happens in the second one? When these chromatids separate themselves in the second process, when they are trying to separate themselves, like for their individuality, like to become the chromatids again, they are separating each other. During that time, during that separation, what happens? The one chromatid from one pair and one chromatin from the other pair, they get crossed over. During separation, they just attach with themselves each other and they get crossed over. So you can see this is known as crossing over. While the maternal and paternal chromosomes are separating, the chromatid material very often gets exchanged between the two members of a homologous pair. This is known as crossing over, which results in genetic recombination. Chiasma is the X-shaped structure formed due to crossing over between the between the non-sister chromatids of the paired homologous chromosome. Now, you have seen that these two chromatids, they pair with each other and they form a X-like structure. This X-like structure is known as chiasma, right? Chiasma is the X-shaped structure formed due to crossing over between the non-sister chromatids of the paired homologous chromosome. That means you can see in the diagram, this chromosome is a paired homologous chromosome. Right, it is a homologous chromosome. Homologous means having the same number of gene sequence, right? So this is having a same number of gene sequence. These are homologous pair of chromosomes. Now when they are trying to separate their chromatids, they get attached to each other and they form a X-like structure which is known as chiasma. And now you will think, what is the use of this? It is written here, this helps in genetic recombination. For recombination or to recombine with each other, this is useful. Now come to page number 18. Yes, this chapter is already over. It is almost over. So this was meiosis. So basically mitosis is a longer process than meiosis. In meiosis, basically the chromosomes are halved. So it will not take too much time, right? So the next page, page number 18, we will read the yellow box. The mixing up of genes during meiotic division provides for the innumerable variations in the progeny, right? When these genes mix up during meiotic division, they varies the number in the progeny. Progeny means the offspring which is formed. You can underline progeny and write over it offspring. So when these mixing up of genes is happening or when it is occurring during the meiotic division, this is causing variations in the offspring, right? So that's why some characteristics of parents are only transferred to the offspring, not all the characteristics, right? We all differ from our parents. That is how the children of some parents, same parents, however similar, are different from each other in certain aspects. It is because of the mixing of genes. When genes mix up during meiotic division, they provide variations in the offspring. That is why we offspring differ from our parents in certain aspects, right? And now the last is a difference between mitosis and meiosis. Just go through it. We all know actually what are the differences. Now this is the basic points where it occurs, what for, when it occurs, everything according to that, on the basis of their occurrence and where it occurs. On the basis of that, mitosis and meiosis is differentiated from each other. So have a look at it after this class. The time is already over. So we will stop here. We will discuss the questions in the next class. Do you all have any doubt in meiosis?
No.
Ma'am.
No? Then in the next class, I will just again repeat what is meiosis and then we will discuss the question and answers, right? So you all may leave. Thank you for today. Okay. If you have no doubt, you may